Okay, hello everyone. Welcome to the first SDB postdoctoral seminar series. My name is Marina Venero, and I'm one of the postdoctoral trainee reps at the SDB board of directors. And I have Krissie Tellez with me today, who is also moderating with me as the second postdoctoral trainee rep in the board of directors. And we're really excited to bring you this new format that SDB is shooting now. And basically, this is a new platform that we're using to present and to highlight the work of our amazing SDB postdoctoral members, especially during these really unique times. And we're really looking to provide them with a platform where they can show us their work, where they can keep in touch with the community, and especially give them a way to present what they're doing now that most of them are going to go into the job market either this year or next year. Krissie, I don't know if you want to add something.
So I'll just give some announcements for the general program. So each talk will be 20 minutes and we'll give a 1-minute warning before the end of the talk. And then we'll have 10 minutes for questions after that. You can enter your questions throughout the session. So we prefer if you write them down as you're thinking of them so they can be answered later in the question and answer chat box. And then we'll read them at the end to the speaker so they can answer your questions. We encourage trainees to ask questions. And if you would like to ask questions anonymously, you can go ahead and do that, or you can have your name. We will be reading the names if not. And then the session will be recorded. So if you need to go back and look at it, you can. We'll give you some more information later on where you'll be able to find that. And this program will be running through May. So we also encourage you guys to keep every month, keep tuning back in. And we really encourage you to advertise this for not only your classes that you might be teaching, but also your labs.
And we're actually really excited because we had more than 400 registered participants today. So that's amazing. Thank you so much for joining. And also we heard that among all these participants, we have actually a group of biology students from Penn State and the Pacific Lutheran University joining us because they're using this platform as part of their biology class. So that's very exciting. I hope you learn a lot today and keep joining us for the next series. And with that, let's get started. So today we have, as Krissie said, 2 speakers. And the first speaker is Michael Piacentino that comes from California Institute of Technology. So Michael is currently a postdoctoral scholar in Marian Bronner's lab, where he studies how neural crest epithelial to mesenchymal transition is controlled by lipid metabolism using CHIC. And Mike completed his PhD in the laboratory of Cynthia Bradham at Boston University, where he was studying skeletal patterning using sea urchins. And his undergraduate studies were at McGill, where he majored in anatomy and cell biology. Mike's interest in developmental biology has led him to participate in several advanced courses, especially the embryology and the zebrafish courses at the MBL in Woods Hole. And he has successfully applied to several fellowships that include one NIDCR K99 Pathway to Independence and the F32 Awards, among others. He has also published several papers showing his work and co-authored many more. So we're very excited to have you, Mike, and we're ready for you anytime.
Wonderful. Thank you so much, Marina, for that really kind introduction. And I want to start just by thanking the Society for Developmental Biology for organizing this really wonderful seminar series to give us postdocs an opportunity to share our work with you. And I'm honored to kick off this series and tell you about how temporal changes in lipid metabolism control neural crest EMT and membrane fluidity. I'm really fascinated by the plasma membrane, which is this highly dynamic structure that encapsulates all of our cells. And I think that this animation illustrates several of the key features of the membrane behavior. We can see that as this plays, the membrane is not a homogeneous mixture of lipids and proteins, but rather is a mixture of more highly saturated lipids over here and some more lowly saturated lipids here. And the thermodynamic properties of these molecules influences the overall biophysical features of the membrane. Among these biophysical properties are things like phase separation, where we can see that the highly saturated lipids will cluster together and partition away from more lowly saturated lipids. And these form scaffolding structures often called lipid rafts. In addition to this phase separation event, we can see that the different saturation states also influence membrane fluidity. For example, the lowly saturated lipids form a much more fluid portion of the membrane compared to those that are more highly saturated. And in vitro and synthetic studies have shown that these biophysical properties go on to influence a number of important cellular processes, and thus understanding how these biophysical parameters influence cell biology in vivo is really at the forefront of disease and developmental biology for me. And so I've selected to study these mechanisms in vivo in a context of high dynamic change, a cellular lifestyle change called epithelial to mesenchymal transition, or EMT. And this is a change where epithelial cells lose those epithelial characteristics and become more mesenchymal and migratory. And the transcriptional and morphological changes that are associated with this EMT event are highly linked to the cellular events at the plasma membrane. In the Bronner lab, we used the neural crest as a model system to study epithelial to mesenchymal transition. And the neural crest is a multipotent stem cell population in vertebrate embryos that arises at the lateral edges of the forming central nervous system, as we can see in these schematics of cross-section through a chicken embryo. During neurulation, the neural crest cells, which are here in red, become localized to the dorsal midline of the neural tube as pre-migratory epithelial cells. They then undergo an EMT in order to gain their migratory capacity and migrate away from the midline. And in this movie, you can see how robust neural crest EMT is. We are looking at the dorsal side of a chicken embryo head, and my colleague Erica Hutchins has labeled neural crest cells with a PAC7 enhancer. And we can see that the cells start out at this midline as pre-migratory and then undergo that EMT to migrate away from the central nervous system. And this EMT is really critical for development, since these cells must migrate away from the midline and throughout the embryo in order to produce a wide range of derivatives that are critical for the final body plan. And so in this system, my central question is, do changes in membrane biophysics regulate this neural crest EMT from epithelial to mesenchymal cell types? And I've chosen to examine plasma membrane fluidity as a jumping off point. And to look at plasma membrane fluidity, I've collaborated with a great friend, Erdinç Sezgin, to use solvatochromic dyes to reveal membrane lipid packing. So these dyes, like NR12S, they integrate into membranes and then they undergo a spectral shift in their emission spectrum, depending on whether they're in a highly rigid or ordered or tightly packed domain in the membrane or a more fluid, disordered domain. And by using spectral imaging, we're able to generate images like this, which is just a giant plasma membrane vesicle that I've phase separated and made this from a chicken cell. And doing the spectral imaging, we can see that there's a domain here where there's high rigidity, and then an opposing domain where there's high fluidity in this membrane vesicle. And so to use this approach in chicken neural crest cells, I electroporated early stage embryos with a fluorescent reporter driven just specifically in neural crest, and incubated these embryos to a pre-migratory stage, at which point I cut out the pre-migratory neural crest and grew them in tissue culture explant scenario and allowed them to undergo their EMT. And when I stain with this NR12S dye and then quantitate the generalized polarization, which is a metric for that fluidity, I see that while pre-migratory cells start with a pretty tight, rigid domain or rigid membrane, EMT and the event of migration then increases the fluidity really dramatically. And so these migratory neural crest cells have a much more fluid membrane than their pre-migratory precursors. So that leads us now to ask, how is it that neural crest cells are regulating this increase in plasma membrane fluidity? How do they go from more rigid to more fluid membranes? And I reasoned that this is likely going to be due to modifications of the lipids in the plasma membrane. And to start to piece together or figure out which candidates those might be, I looked at RNA-seq data published by Williams et al. Last year, and I reanalyzed this data and parsed out genes that have lipid-associated Go terms. And thus reasoned these might be metabolic genes or genes that are affecting membrane dynamics. And by doing a heat map clustering analysis, I see that there's a set of genes that are enriched highly in the pre-migratory neural crest cell stage, and then another set that is enriched in the migratory crest. And so I imagine that these genes may be playing a role in regulating this increase in membrane fluidity. And so for the rest of this talk, we're going to focus on one of these genes called SMPD3 that encodes a protein called neutral sphingomyelinase 2, or NSMACE2. NSMACE2 is, here I confirmed that it's expressed within the neural crest cells at the time points of EMT. Its expression initiates at pre-migratory stages here in this earlier stage chicken embryo head, and that expression level maintains throughout migration. Biochemically, neutral sphingomyelinase enzymes cleave phosphocholine off of sphingomyelin to produce ceramide. And both of these are highly saturated and often long acyl chain lipids that reside in the plasma membrane. And so by understanding what NSMACE2 is doing, we're testing what the function of the production of ceramide in the plasma membrane is. And I just went a little bit further to confirm that ceramide, the product of NSMACE2, is indeed present in the migratory neural crest cells in this cross-section of these migrating crest cells that are labeled with PAC7. We can see that there's enrichment to this antibody labeling along these cells. And so I was asking then if NSMACE2 is necessary for plasma membrane fluidity. And so I repeated this NR12S spectral experiment, but used neural crest cells that I electroporated a knockdown reagent, an NSMACE2 morpholino. And when I quantitate the polarization state of these membranes, we see that the pre-migratory morphant crest cells are similar to the pre-migratory control. However, the NSMACE2 deficient migratory crest have a much more rigid membrane than those from the control scenario. And so this NSMACE2 is really necessary to induce this membrane fluidity change. And so now I wanted to ask, what is the role of NSMACE2 and membrane fluidity during the overall development of neural crest cells in vivo? And so in chicken, we have this remarkable system where we can inject either side of the embryo with a control construct or a knockdown construct. And as we develop these embryos, then the neural crest gets bilaterally segregated into the control on one side and the knockdown crest on the opposite side. And if you don't believe me that there's this clean line down the middle, here's just GFP and RFP electroporated on either side. And so we've got this beautiful system where we can have this perfectly stage matched contralateral control. And so all of my images of embryos are going to show scenarios like this, with SMACE perturbation on the right and a control on the left. And so if I knock down NSMACE2 and develop two migratory neural crest stages, we see that the control side shows a nice wave of migration out of the chick head. However, the SMACE deficient neural crest cells fail to undergo that migration, and it looks like they're clumping up and failing to undergo their EMT. And we can quantitate this migration area as shown here, and there's really a significant block to neural crest migration. I then went a little bit further and I looked at some canonical EMT markers in neural crest cells, such as SNAIL2, which promotes EMT, and CAD6B, which is a cadherin molecule expressed in pre-migratory neural crest cells that maintains adhesion and therefore is kind of anti-EMT. And when we immunostain for SNAIL and CAD6B, we see that SNAIL expression levels in these neural crest cells are reduced when we lose NSMACE2, and CAD6B remains pretty high. And this indicates that SMACE or NSMACE2 function and ceramide production are necessary for EMT and for the release of these cells from their neighbors or their de-adhesion. I went a little bit further and looked at the pre-migratory and migratory, or I looked at the neural crest gene regulatory networks using hybridization chain reaction. And in pre-migratory neural crest cells, we normally expect MSX expression and CAD6B expression. During EMT, SOX9 and SNAIL2 turn on to repress CAD6B expression. And in NSMACE2 deficient cells, we see that these early genes are not expressed while CAD6B remains highly elevated. And so I wanted to know, how is it that this plasma membrane activity of NSMACE2 is changing these transcriptional profiles in the nucleus? And we know that the initiation of these gene expression profiles is downstream of several canonical signaling pathways, including Wnt and BMP. And so I reasoned that NSMACE2 may be having a role at the plasma membrane to regulate Wnt and BMP signaling. And so I asked what's happening with these two pathways. And to test Wnt and BMP signaling, I used a transcriptional reporter for both of these pathways that drive GFP expression. And when I quantitate the activity of a Wnt sensitive or a BMP sensitive reporter, we see that both Wnt and BMP pathways are downregulated, indicating that NSMACE2 is required for both Wnt and BMP signaling. And so I'm going to start building up this model where we have pre-migratory neural crest cells that may be rich in sphingomyelin in their membranes. And then at the onset of EMT, NSMACE2 converts this to ceramide, which then somehow goes on to promote de-adhesion, the release of cadherins, and signaling through Wnt and BMP pathways. And so I next asked, how is it that membrane lipids could be controlling these signaling pathways? And our logic for this comes from the observation that endocytosis actually potentiates a lot of signaling pathways, and that's shown to be critical for Wnt signaling, but also for BMP, where these activated signaling complexes become internalized and processed into multivesicular endosomes. On the other hand, ceramide production in vivo or in vitro scenarios and in synthetic scenarios has been shown to really potentiate membrane curvature and internalization of highly rigid membrane domains. And so this led me to ask if endocytosis is the mechanism by which NSMACE2 regulates neural crest EMT. And so I tested this using a receptor or an endocytosis assay using transferrin or TF633, where I bathed transfected epithelial cells with this reagent and then quantitated how much endocytosis occurs. And if I look at just an RFP control, I've plotted the distribution frequency of endocytic puncta like this. And what we really need to know from these graphs is if the curve that we trace shifts to the left, that will indicate that endocytosis is reduced. And if it shifts to the right, it'll mean endocytosis is increased. And so I did a little control where I inhibited endocytosis by overexpressing a dominant negative dynamin 1. And we see indeed a reduction in the transferrin internalization. Surprisingly, if I overexpress NSMACE2, we get this really significant increase in transferrin internalization, indicating that NSMACE2 is sufficient to induce endocytosis. And finally, if I mutate the catalytic domain of NSMACE2, we see no change from a control. And that indicates that it's the actual catalytic function and the formation of ceramide by NSMACE2 that is necessary for endocytosis. And so I'm left now asking, can endocytosis explain the NSMACE2 loss of function phenotype? And I tested that by driving expression of the dominant negative dynamin construct, specifically in cranial neural crest cells. And we see that compared to the control, there is also a significant reduction in the migration of neural crest. And if we compare that magnitude to that of the NSMACE deficient cells, we see pretty much a phenocopy where inhibiting endocytosis phenocopies the effect of NSMACE2 loss. And further, we've shown that inhibiting endocytosis in CREST also reduces Wnt signaling comparable to what we saw with NSMACE2 loss of function. And so now we can take this model and add a little bit more complexity. And by the production of ceramide, this is introducing membrane curvature that is then encouraging endocytosis and then de-adhesion from neighbors and cell signaling. And so ceramide is really required for neural crest EMT. But we also saw at the beginning of this talk that during migration, the plasma membranes have increased fluidity. And this is likely, or this is my hypothesis, is that the endocytosis of these highly saturated lipids is leaving behind a membrane of increased fluidity. And so to investigate what fluidity is doing during migration, I did a quick little pilot experiment where I time-lapsed migration of control and NSMACE2 deficient neural crest cells. And I see that while control cells undergo a nice, what looks to be a directional migration, the NSMACE2 deficient cells fail to move very much. And if I trace their nuclear positions for a large number of cells over time, we see that the control morpholino cells have this exponential increase, which is consistent with a directional migration, while the mean squared displacement for NSMACE2 deficient cells is more linear, which is consistent with random walking behavior. And so what I've shown you today is that during neural crest EMT, we have this conversion of sphingomyelin into ceramide under temporal regulation by the expression of NSMACE2. And that ceramide then promotes membrane curvature and endocytosis. And this endocytosis enables for de-adhesion from neighbors and important cell signaling events that allow for transcriptional changes and activation of the migratory program. And finally, that endocytosis then increases plasma membrane fluidity and somehow is affecting directional migration. And so I find it really amazing how one subtle lipid modification can initiate a long chain or a long-reaching cascade of cellular events that are necessary for EMT. And by using the chicken neural crest system, we're now able to test hypotheses on membrane function that have been generated from in vitro studies, but do so in an in vivo context. And so going forward, I'm really excited for my work to continue to expand on how plasma membrane fluidity drives directional migration. And in addition, I'm curious how the content, the sphingomyelin and ceramide content at these earlier stages influence phase separation and the formation of those lipid rafts, and how that phase separation partitions proteins at the membrane. And these proteins could be our signaling complexes or cadherin molecules during adhesion. And finally, I've shown you this mechanism that occurs in chicken embryos, which develop at 37 degrees Celsius. And since the properties of lipids are highly dependent on the temperature, I'm excited to explore how this mechanism is conserved between different species. And if I look at other species such as zebrafish that develop at cooler temperatures, I want to determine if these different organisms use the same mechanism or if they co-opt a different set of plasma membrane lipids in order to activate the same goal of EMT and migration. And so these are the general areas that I'm hoping to take my research. And with that, I would love to thank my advisor, Dr. Marianne Bronner, for all of her support in investigating this really exciting and sometimes challenging project, as well as support from the whole lab who have provided a lot of input on this process, and especially Erica, Krissie, and Alexis, who've contributed to the actual production of this data. I also want to thank Megan for, Stephen and Justin for help with analysis and interpretation, and support from the imaging facility at Caltech. And finally, collaborators such as Erdinj Sezgin and Tatiana Sauka-Spangler at University of Oxford for help with some of these fluidity measurements. I'd also like to thank support from funding agencies and for SDB, And finally, thank you all for attending today, and I'm looking forward to any of your questions. And if we don't have time for those questions, or if you're watching this later, feel free to reach out at my email address here or by Twitter, and I would love to start a discussion.
Great.
Thank you all.
Oops, sorry. Thanks for such a beautiful talk, Mike. So let's head to the question. So the first question is, does NMACE2 knockdown also affect the apical basal polarity of the cells and their junctions?
That is an excellent question. In epithelial cells, the apical membrane is really highly rich in the long-chain saturated lipids, such as sphingomyelin and ceramide. And the partitioning of those lipids to the apical surface in epithelial cells compared to where those partition during migration in mesenchymal cells, I'm not entirely sure yet, and I'm really excited to see if NSMACE2 activity is playing a role in that polarization. There are bits of evidence that suggest that it does help polarize G protein activity and migration. And so whether it is by positioning the lipids or some other protein components is really an exciting future direction.
Okay, so Xiao Wu Gu, I hope I didn't mess up your name, asks, very cool. Have you looked at the pre-migratory and migratory cells ultrastructurally? I wonder how caveoli are different there. Also, have you performed lipidomics on these cells?
Both excellent questions. I'm starting to dig into the caveoli work now. Their caveoli tend to be rich in sphingomyelin and need to be destabilized, and the removal of sphingomyelin from those caveoli will just destabilize them and can allow them to be then internalized from the membrane. And so I haven't done any ultrastructural experiments just yet, but that is definitely a future direction that we're seeking to go. As for lipidomics, I love that idea, and I really want to do lipidomics, and I just have not gotten around to it yet because it is particularly challenging to isolate a sufficient sample size with these cells. We only get a couple hundred per embryo, and we need 10 to the 6th, 7th, 8th for bulk mass spec lipidomics. And I'm a little bit worried about dissociating these cells, how that's going to change their membrane lipidomes. So if anybody has advice or experience with this, please drop me a line. I'd love to chat.
Thank you. So Daphne Schatzberg asks, how can you tell the NMACE2 isn't affecting mobility rather than EMT? Could knockdown cells also be undergoing EMT but failing to move?
That is definitely a good question. And I think that what this is doing is actually playing this bimodal function where the production of ceramide is triggering transcriptional responses that are necessary for EMT. And so I think that that is one feature that's before the migratory program. And then later on, when I culture these cells outside of an embryo where they aren't as confined or need to complete such a dramatic EMT, we do see occasional migration and recovery. And so I think that it is playing a role both during EMT to activate this program, but then later to help for polarization and directional migration. And teasing apart those two functions is a fun challenge I'm looking forward to.
Sorry. Thank you. So Melissa Trong also asks, really cool, is overexpression of NMACE2 sufficient to drive early migration / EMT?
That is definitely what I had hypothesized. I tried that experiment and it does not quite work. I think that the problem with this type of experiment is that if we are overactivating endocytosis prematurely, those receptors on the cell membrane might not have had a chance to bind their signaling ligands. And so if you're endocytosing an empty receptor, you're not going to activate your signaling pathway. And consistent with that, when I overexpress MACE prematurely, I also block migration. It's as if they fail to turn on their EMT program because they failed to have that signaling event happen.
So an anonymous person asks, have you had a look already at how conserved NSMACE is across species?
Yes, I have sort of hinted at future directions there, but for example, they do not express, their neural crest cells do not express these NSMACE2 genes. And I think that is because sphingomyelin and ceramide are so rigid as lipids. They're very long chain, very saturated. And at cooler temperatures, that saturation state might be just too much to overcome their rigidity. And so I hypothesized that this whole downstream curvature endocytosis cascade is conserved in zebrafish, but it's a different set of lipids that might be less saturated that would activate this cascade. And so I'm digging into expression profiles to look for what these lipid modifying genes might be and what those lipid candidates could be.
Thank you so much. All right, in the interest of time, the rest of the questions, I will dismiss them and Mike will be able to type an answer if he so wishes to do so, or you guys can email and reach out. So I'll dismiss them and you can respond that way. But thank you so much, Mike.
Will do. Thank you.
And then we're gonna go ahead and set up for the next speaker, who is Natasha O'Brown. So I'll just read her introduction as she's setting up. Oh. So Natasha is a Damon Rennion postdoctoral fellow in the labs of Jinghua Gu and Sean Megason at Harvard Medical School. She is uncovering the molecular and subcellular basis of the blood-brain barrier development in zebrafish. So Natasha completed her PhD in developmental biology at Stanford University, where she worked in the lab of David Kingsley. So in that lab, she investigated the molecular basis of human and stickleback evolutionary adaptation. And for undergraduate studies, she majored in biology and graduated magna cum laude. Natasha has been recognized by several awards and fellowships, including the Damon Rennion Postdoctoral Fellowship, Dale F. Frey Award for Breakthrough Scientists, and she has presented and published her findings extensively, and also serves as a mentor for young stage scientists. So we're happy to have her with us today, and we're excited to hear more about her work. So take it away, Natasha.
Okay, hi, you guys hopefully can see the right thing this time. So like she said, I'm Natasha, and I'm gonna be talking to you today about my studies using zebrafish to understand the neurovascular interactions that regulate the development of the zebrafish blood-brain barrier. Oops. And so the brain is the most densely vascularized organ in the human body. It requires, has an extremely high metabolic demand that requires tightly linked neuronal and vascular patterning that happens during development. It also requires a rapid blood flow response to increase neuronal activity, and this is called neurovascular coupling. And lastly, it requires tight regulation of what enters and exits to maintain proper neuronal function, and this is achieved via the blood-brain barrier. So the blood-brain barrier, or the BBB, is the set of properties that's unique to the vasculature of the brain, here marked in green. And when we zoom in, we can see that the blood vessels are composed of a single thin layer of endothelial cells, here marked in green. And these share a basement membrane with these mural support cells called pericytes, here in purple. And then in the mammalian systems, the entire vasculature is completely ensheathed by astrocytic processes or astrocytic end feet in blue. And so the BBB controls which molecules enter and exit the brain. It maintains ion and nutrient homeostasis, and it protects the brain from toxins and pathogens. And while in day-to-day life, the BBB acts benevolently to help protect the brain, it's also a huge obstacle for drug delivery in disease states. And so when we zoom into these endothelial cells, we can see that unlike peripheral endothelial cells depicted here in green, brain endothelial cells have two key characteristics that create this restrictive barrier. So first, they express specialized tight junction complexes between neighboring endothelial cells. And these block the passage of small water-soluble molecules from the lumen between cells and into the abluminal space. And the second thing they have is reduced rates of vesicular trafficking or transcytosis across the cells. And through a series of classic chick quail transplant studies done by Stewart and Wiley in the 1980s, we now know that these specialized properties are not intrinsic to endothelial cells, but rather are acquired from signals in the brain microenvironment during development. However, we still don't know which cells are producing these and what those signals are. And so while there's a growing list of hallmarks of BBB endothelial cells from the rapidly booming RNA-seq data that's coming out, like tight junction molecules, Claudin-5, ZO1, LSR, influx and efflux transporters, as well as the fatty acid transporter MFSD2A, which explicitly blocks transcytosis in brain endothelial cells, there's still a lot of big questions that are remaining in the field, such as when and how does the BBB become functional during development? And then what are the dynamics of BBB leakage, both developmentally and in disease states? And then which cells in the brain microenvironment regulate these endothelial cell properties, both to induce barrier properties during development, as well as to maintain barrier properties in adulthood? And lastly, can we identify molecules that regulate BBB permeability to either help improve therapeutic accessibility to the brain or to help restore barrier properties in neurodegenerative diseases? And so while the vast majority of BBB studies rely on mouse models, I instead have turned to zebrafish, because zebrafish offer a powerful tool to study development in vivo, as many of the people in the audience already know. And as you can see from Ian's beautiful video, zebrafish develop rapidly and externally in the span of a few days. They're optically transparent and small enough so you can actually image entire tissues in vivo. And not only are they the perfect size for imaging, but each cross generates hundreds of eggs versus the 8 to 12 pups that you can get with mice, which is why they've been used for toxicological and drug screens for decades. And now with the advent of CRISPR, we can also easily genetically target and mutate specific loci within the genome. And so while all of this shows the beauty of zebrafish as a developmental model system, you might be wondering whether or not zebrafish even have a blood-brain barrier. So in fact, they do. I wouldn't be here if they didn't. So as you can see here in this sagittal, so this is a side view cut through the brain. And you can see that this serum protein, it's a transgenic protein, 80 kilodalton DBP EGFP, stays confined within the vasculature throughout the forebrain and the midbrain. Not only do the adult blood vessels confine tracers within the brain, but these endothelial cells, here marked in green by EGFP, they also express similar tight junction proteins like ZO1 and Claudin-5 here in red. So while all this suggests that they're a great model for blood-brain barrier, we're left with the question of when does the barrier form in zebrafish? To address this, I used a transgenic line of zebrafish that expresses M-cherry throughout the entire vasculature under the KDRL promoter. And then by injecting these fish with fluorescent tracers into the heart and then allowing that tracer to circulate throughout the body for an hour, and then doing this at different developmental stages, I can see when the tracer becomes excluded from the brain, indicating that the blood-brain barrier had become functional. So here is a representative top-down view now of the entire larval zebrafish head at 3 days post-fertilization, or DPF. And you can see that the tracer is in turquoise and the vasculature is marked in magenta. And as you can clearly see, just to orient you, sorry, the forebrain is pseudocolored in gray here to the left, the midbrain in violet, and then the hindbrain in gold. And so you can clearly see, using this tracer leakage assay, that throughout the brain, the tracer is making its way into the brain parenchyma outside of the blood vessels. And when we focus in on the midbrain for the sake of time in this talk, and we do this with different tracers, we can see that both the 1 kilodalton NHS in turquoise, as well as this 10 kilodalton dextran in white, are extravasated out of the blood vessels and entering the brain parenchyma. And this is true also for the transgenic serum protein, so this isn't just some sort of weird injection artifact. When we repeat this at 4 days, we similarly observe tracer uptake in the brain parenchyma for all 3 of these tracers. However, by 5 days, hopefully you can notice that there's a rapid reduction in the amount of tracer that we see outside of the blood vessels. And this becomes more apparent when you actually look at the quantification across many fish, that basically between 3 and 4 days, there isn't much difference. And then there's this rapid fall off at 5, and this stays pretty consistent up until 10 days post-fertilization. So now that we know when the BBB becomes functional in the fish, we next wondered what the subcellular mechanism was driving this leakage. Is it through tight junction function, or is it through transcytosis? And so to do this, I performed similar tracer leakage assays, but this time with electron-dense gold nanoparticles followed by electron microscopy rather than fluorescent tracers. And so as you can see here at 3 days post-fertilization, the mural support cells, pericytes, are already in close contact with the endothelial cells, pseudo-colored in green. However, what we did notice was that the electron-dense tracer, which is black under EM, it's filling up the endothelial basement membrane, which is what all of these turquoise arrowheads are kind of blaring at you for. And when we zoom in, we can see that this is not occurring through tight junction function, as the tracer is halted at the so-called kissing point between endothelial cells, but rather we see an increase in these numbers of these small flask-shaped vesicles marked by the pink arrowheads. When we repeat this at 5 days post-fertilization, when the barrier is functionally restrictive, we notice that the basement membrane is pretty devoid of this gold tracer, and this coincides with a reduction in the number of these small vesicles. And this stayed true up until 7 days post-fertilization. And this reduction in the amount of basement membrane gold intensity occurring at 5 days, as well as vesicular abundance, is quantified here in 4 different fish with 10 vessels per fish. So now we know when and how the BBB is developing and becoming functional in the fish. We get to do some of the fun experiments, which is trying to figure out how the BBB leakage is occurring in vivo in an intact animal. So now I injected a tracer in green. I showed a teaser of this on Twitter so you can see that in this 3-day-old fish head that the tracer is filling the brain parenchyma. You can also notice that up here in this corner, there's active angiogenesis that's also occurring as the blood is flowing through. And hopefully you can just appreciate, just the full beauty of being able to actually image these things in a live organism. And so when we zoom in, I'm now going to highlight cells that are taking up tracer by these white lines and kind of pausing. But we observed 2 different types of tracer accumulation. So first we observed what we term this large bursts, where the nearby cells are taking up tracer in under a minute and to equal levels of what is actually in the vessel lumen. And then in addition to these bursting activities, we also saw an accumulation of this diffuse dextran uptake in the brain parenchyma. So this kind of speckles that you start seeing accumulate in the background. However, when we repeated this at 5 days post-fertilization, we didn't see either type of leakage happening. There wasn't nearly as much of the diffuse tracer accumulation, and we never saw any of that bursting activity. And so when we quantify time 0 to the final time in either 3-day embryos and 5 days, hopefully it's very obvious that the amount of accumulation at 3 days is way more than at 5 days. And then to actually understand the dynamics, we measured the tracer intensity in 6 different regions throughout the brain parenchyma, marked by these yellow boxes, and then did this over time and normalized it to the luminal tracer intensity to account for fish-to-fish variability, and did this in many, many fish. And so when we see that, we can see that while at 5 days there's a slight increase towards the end of the hour of imaging, the 3-day-old fish have this steady and gradual increase in tracer uptake in the brain parenchyma that starts almost immediately. And this is some of the first data on what is actually occurring developmentally in terms of BBB permeability. Oops. And so now that we have this system set up, we can actually start to tease apart how the various aspects of subcellular properties of brain endothelial cells regulate BBB permeability dynamics. And so we focus primarily on transcytosis because we know from extensive mouse data from the GU lab that MFSD2A mutants specifically increase caveoli-mediated transcytosis in brain endothelial cells. So with this in mind, we generated MFSD2AA mutants, which is the MFSD2A ortholog in zebrafish. And these MFSD2AA mutants display increased tracer leakage at 5 days post-fertilization, as seen here on the right. And this increased permeability is occurring due to an increase in small flask-shaped vesicles, as marked here, compared to their wild types, just as observed in the mice. So as you can see here, on the left, the heterozygote controls are not extravasating the tracer that was injected, and so it's as expected. However, the MFSD2AA mutants do show an increase in tracer uptake, and you can see that it's not happening in any of this bursting activity, but rather this kind of gradual diffuse spread of tracer. And so when we quantify the tracer intensity over time in several mutants versus their heterozygote controls, you can appreciate that MFSD2AA mutants have this increased steady uptake of the dextran, whereas the heterozygote controls look exactly like the wild type 5-day-old embryos. And in fact, when you do the side-by-side comparison of the tracer uptake in 3 days versus versus the mutant 5-day uptake, you can see that they're happening at about equivalent levels and to the same degree, suggesting that this early leakage is happening primarily due to increased transcytosis that we observe from the electron microscopy. Okay, so during the course of these developmental BBB studies, I stumbled upon a novel mutant. It was kind of a pain in the ass because it kept appearing in all of my CRISPR strains, but it's a beautiful mutant, at least in my eyes, it's beautiful. So this leakage is far more impressive than the leakage that I observed in the MFSD2AA mutants, but it's also much more regionally restricted. So these mutants are specifically leaky in the forebrain and the midbrain, whereas their hindbrain is relatively wild type. And it's important to note that while impressive with these fluorescent tracers, this isn't brain hemorrhage, so I'm not seeing blood pooling. And these fish are completely viable and they look totally normal by eye until you put them under the confocal microscope. And so the other thing to mention is that while there is all of this vast leakage, their vascular patterning remains unaltered. So their vascular density and branching is unperturbed, as well as their parasite coverage has remained the same. Okay. So in order to identify what gene is actually causing this mutation, I used RNA sequencing and linkage mapping. And this led me to a single peak on chromosome 14, marked in red, and more specifically to the SPOC1 gene. So SPOC1 encodes a secreted chondroitin sulfate and heparin sulfate chimeric proteoglycan with 3 discrete protein domains. So it has this Casal serine protease inhibitor domain. It has a Spark extracellular calcium binding domain, and then this thyroglobulin cysteine protease inhibitor domain, as well as being decorated by these glycosaminoglycan side chains. And while the RNA sequencing didn't reveal a difference in SPOC1 expression between mutants and wild types, it did reveal a single missense mutation T241A that falls within the Spark extracellular calcium binding domain. So to validate that loss of SPOC1 activity is indeed responsible for this leakage phenotype, I used CRISPR, and instead I used 4 different guides here marked by green, targeting various loci within the SPOC1 gene, and then assessed tracer leakage within the mosaic F0 crispins. And so while uninjected controls look as expected, I saw a range of phenotypes in the crispins. And so there were several of the crispins that completely phenocopied the leaky mutants, here like crispin number 2, which indicates that indeed SPOC1 function is necessary and responsible for the leaky mutant phenotype. However, my personal favorites were the handful that actually displayed this hemisphere-specific leakage pattern. Like I said, these are mosaic crispins, which suggests that somehow localized SPOC1 activity is necessary and sufficient for driving these barrier properties, and raises some interesting perspectives. So now that we know that SPOC1 is the gene that's causing this leakage phenotype, we next want to know, where is SPOC1 expressed? So I use, okay, thanks. SPOC1 HCR in situs, and I can see that there is fluorescent signal, in the brain and the retina, so throughout the CNS. And this expression coincides with the neuronal signal ELA-VL3, but not the vascular signal KDRL. And this is even clearer here in the zoom in, where the vascular signal is outlined in white. You can see that SPOC1 is completely absent and is mostly co-occurring with the neuronal signal, which is what is observed in mouse and human. So the next question is, what's the subcellular mechanism of this? So we turn to EEM and again see that the neurovascular unit is basically the same. And while the vast majority of the tight junctions were functional at this point, 88%, we did notice an increase in these small flask-shaped vesicles with the pink stars. But in addition to those, we also noticed these large vesicles that are greater than 200 nanometers in diameter that I've never seen in any other mutants, and this is probably why the leakage is happening faster than the MFSD2A mutants. And this increase in both types of transcytosis is quantified here. Okay, so lastly, we want to know whether or not this was just some weird fish thing or if it was conserved to mice. And so SPOC1 is also known as testican1, and the knockout already existed. So in the wild type embryonic day E15.5, they're able to confine the tracers within their blood vessels, as seen here with NHS biotin and the 10 kilodalton dextran. However, the mutants do not, suggesting that that SPOC1 is playing a conserved role, and this isn't just a one-off. This is true for 4 littermate controls in 3 different litters, suggesting that this isn't some weird fish thing and that SPOC1 is playing a conserved role in determining barrier properties. So back to the big questions in the field. I hope I've shown you that the zebrafish BBB becomes functionally restrictive by 5 days post-fertilization, specifically through the suppression of transcytosis. I've shown you that we can actually image and perturb endothelial cell properties and see how they affect the dynamics of BBB leakage. And that we've just begun to scratch the surface on which cells in the brain microenvironment and which molecules play a role in determining BBB leakage by identifying the first neuronally produced signal, SPOC1, that regulates BBB properties. And I hope to uncover many more when I'm able to start up my own lab using this powerful system that I've been able to establish during my postdoc. And with that, I'd like to acknowledge the GU and Megasin labs, and especially my funding with Damon Runyon, and take questions, and I will try my best to address any questions I can't answer live on Twitter, so look out for that. With that, I'd like to just say thank you.
Thank you, Natasha, that was great. So let's see, we have some questions for you here, and I'm combining some because they might be overlapping a bit. So you mentioned that your zebrafish work entirely has been done in vivo live imaging, right?
Yeah.
Okay. And Mia Konjikusik is asking, she's saying, beautiful work. I was wondering if you can see how quickly your tracer is cleared from the CSF or ventricular spaces.
Yeah, I honestly haven't done that much imaging of the clearance, but it does stay for at least a few hours. Definitely by 24 hours, the injected dextrans and stuff like that are gone, and those just accumulate in the liver.
Okay. Junlu Zhu is asking, since microglia are already active by 3 to 5 days post-fertilization, did you see microglia carrying tracers crossing the BBB?
I definitely do see microglia. Some of those cells in the earlier images that are taking up the tracer are definitely microglia.
And Kara Cerveni is asking, really beautiful work. Given the expression pattern of SPOC1 in both midbrain and retina, did you notice a correlation between BBB and BRB maturation? Do you know if fish with a leaky BBB also have a leaky BRB?
So I honestly haven't been, I'm not an expert in BRB development, and so I haven't actually been looking to see whether or not the BRB is happening at the same developmental stages as the blood-brain barrier. But I do think that SPOC1 mutants do have blood-retinal barrier leakage as well. So.
Awesome, thank you. Jesus Torres Vasquez asks, do you see increased lymphatic vessel growth in your mutants?
I have not looked at that explicitly.
I'm curious too.
Yeah.
And Manideep Kavali is asking, do Spock mouse mutants display the same forebrain, midbrain, BBB defects as you see in zebrafish?
Yeah, so they seemed less regionally restricted. And I'm starting to get into actually potentially part of what I didn't get to show you is that this phenotype is happening between like the parasite interactions with the endothelial cells is not the same. And so in the fish, the forebrain and midbrain parasites are neural crest derived and the hindbrain ones are mesoderm derived. And in the mouse, I think more of the parasites are more like intermixed throughout the brain. But I think that there's a neural crest aspect of this that's coming out.
I see. Let's see, Julie Siegenthaler asks, awesome work. Neuronal SPOC1, any evidence from the literature how it could be altering transcytosis in the endothelial cells?
Yeah, so there is really like zero evidence in terms of function of what SPOC1 is doing. It's been correlated with a lot of oncogenic activity and EMT actually, so we're going all the way across. But basically, when it comes down to like what SPOC1 is actually doing, that is what my prime directive for the next year is basically trying to understand how it's connecting to the endothelial cells.
Okay, let's see, maybe one more, we have some time. Let me see, this is more. Selena Valvano is asking, I may have missed this, but what is MFDS2A's role and what does it normally uptake? And I will complement that with where it's actually expressed. Is it expressed in vessels?
Yeah, so MFSD2A is a fatty acid transporter. So it's a lipid flipase that flips into the membrane. And normally, basically what it's doing is by changing the lipid membrane composition, which plays in nicely to Mike's talk, it suppresses caveoli vesicle formation in brain endothelial cells and actually spinal cord as well. So,
fantastic. Yeah. And I think we have 1 minute for the last one. I'm going to take the last. Scott Hawley is asking, are the SPOC defects restricted to the brain vasculature or the leakage is everywhere, elsewhere in the fish?
Yes, I mean, the rest of the body of the fish is normally leaky, so I don't see anything that's extra leaky. Yeah. But I mean, it's definitely not, it could have other impacts in other places. So SPOC1, at least by antibody and stuff, it seems like it does build up in the jaw and some other neural crest tissues. So like, I wouldn't be totally surprised if there were some other neural crest defects that are happening.
Okay, we have more questions, but we unfortunately are running out of time. So we'll send you those so you can answer them maybe. But with that, really, we want to thank you. And we want to thank Mike for your great talks today. It was great listening to your research. Thank you everyone who came to the talks, and we hope to see you in a month. And actually Natasha and Mike are gonna be the moderators for the next session in November, so we'll have them back here. Thank you so much, guys. Thank you everyone for coming, and we're here for you. So if you need us, please reach out and tweet.